Good day, everyone. So today we're going to talk about mitosis and meiosis. So in this video, we're going to talk about the different stages of mitosis and meiosis and what is their difference. So what is mitosis? Mitosis is a process of cell division where chromosomes are replicated and separated into two genetically identical cells. Mitosis is also the process involved in growth and repair of our body. So what are the process of mitosis? So right now we're going to tackle the different stages of mitosis. So this is the phase wherein duplication of nuclear DNA takes place. The first stage is interphase. Interphase is the phase wherein the cell grows, duplicates its organelles, and most importantly, replicate its DNA. The next stage is prophase. So prophase is the stage wherein the chromatin condense in the visible chromosomes, and then the nuclear envelope begins to break down and the spindle fibers began to form. So the prophase is the stage wherein the chromatin condenses, meiotic spindle assembles, and the nucleus disappear. So the next stage is metaphase. So the stage of metaphase is the phase wherein the chromosomes align along the metaphase plate. So this is the part wherein the chromosomes align on the equator of the cell. So this is also the part wherein the spindle fibers attach to the kinetochores on each sister chromatid. So this is the place wherein the spindle fibers is attached on the chromatid sister and the poles of the cell. So next is anaphase. So anaphase is the phase wherein the sister chromatids separate and are pulled apart by the shortening spindle fibers toward the opposite poles of the cell. So this is the phase wherein the sister chromatid is being separated and the other one is being pulled to the other pole and the other one is being pulled to the opposite pole of the cell. And that is because of the spindle fibers that is shortening and going towards the opposite poles of the cell. The next one is the telophase. So this is the phase wherein the chromosomes arrived at the poles and begin to decondense. So in this phase, the nuclear envelopes form around the two sets of chromosomes and the spindle fibers disappear. So this is also the phase wherein the two sister chromatid is now at the poles. So now we've come to the last stage, which is the cytokinesis. So in this stage, the dividing of cytoplasm and the rest of the cell contents into two distinct daughter cells happen. So in this case is the part wherein a new cell is being formed. The cytoplasm is being divided into two parts and the other one is one cell and the other one is the daughter cell. So that is the 5 stages or phases of mitosis. So the first stage is the duplication of nuclear DNA takes place. The second is the chromatin condenses, mitotic spindle assembles, and the nucleus disappears. The third stage is the chromosomes align in the middle of the dividing cell. And the fourth stage is the spindle fiber splits apart the chromatids to opposite poles of the cell. And the last but not the least phase is the phase wherein the new sets of identical cells are generated. The nucleus starts to form around each new cell. So again, always remember the 5 stages of mitosis.
Now, let's try to tackle about the topic of meiosis. Meiosis is the type of cell division that produces 4 cells with half the number of chromosomes from the parent cells. This process is responsible for producing egg cells and sperm cells. Meiosis is also divided into 5 phases. The first one is prophase 1. During this stage, the DNA replicates, resulting in chromosomes made of 2 sister chromatids. The second phase is the metaphase 1. Homologous chromosomes pair are aligned along the center of the cell. The third one is anaphase 1. Homologous chromosomes separate and are pulled to opposite poles of the cell. Sister chromatids remain attached. And the fourth phase is telophase 1. The nuclear envelope may reform around the separate chromosomes at each pole. And the last one is cytokinesis. Divided the cell into two genetically unique haploid daughter cells. And that's the 5 parts of meiosis 1. Tetrads are formed, chromatids exchange segments of allies resulting in different genes combination. The nuclear disappear, the chromosomes align in the middle of the divided cell, spindle fiber starts to form. The homologous chromosomes are split and pulled towards opposite poles of the cell. Two new sets of non-identical cells are generated. The nucleus starts to form around each new cell, dividing the cell into two genetically unique haploid daughter cells. Also, meiosis have second part. It has a prophase 2 where the nuclear envelope breaks down and chromosomes are recondensed. Also, it has metaphase 2 where individual chromosomes align along the equator of the cell. Also, we have an anaphase 2. Sister chromatids separates and move to opposite poles of the cell, becoming individual chromosomes. And then we have telophase 2. Nuclear envelopes reform around each set of separate chromosomes at the poles. And last but not the least, we have cytokinesis 2. It results into 4 genetically unique haploid daughter cells. And that's all the 4 phases of meiosis 2. Spindle reform around the chromosomes of each new cell. The next one, the nucleus disappear. The chromosomes align in the middle of the dividing cell. The next one, the sister chromatids are split and pulled towards opposite poles of the cell. And lastly, 2 new sets of non-identical cells are generated from each divided cell. The nucleus starts to form around each new cell. But where do mitosis and meiosis occur in our body? Mitosis occurs in almost all somatic, or which we call body cells for growth, repair, and cell replacement, while meiosis occurs in testes in male and the ovaries in female to produce gametes, which we call sperm and egg cell.